On June 5, 1991, NASA launched one of its most unusual missions aboard the Space Shuttle Columbia. This was STS-40 carrying Spacelab Life Sciences 1, and it would make history as the first shuttle mission devoted entirely to studying living organisms in space. The crew of 7 astronauts included Commander Brian D. O'Connor and mission specialist Rhys Seddon, who would help conduct experiments during the 9-day flight orbiting at 157 nautical miles above Earth. Getting to that launch pad had not been smooth sailing. The mission was originally scheduled for May 22, but problems emerged less than 48 hours before liftoff engineers discovered a leaking liquid hydrogen transducer in the orbiter's main propulsion system. This component had been removed and replaced during leak testing the previous year, but the vendor's analysis showed it had failed. The team also faced issues with computer systems. These complications pushed the launch back by nearly 2 weeks. The delay was frustrating for everyone involved, but the science was too important to rush. This mission represented a major investment in understanding how life responds to the space environment. Among the many experiments on board were those jellyfish polyps waiting in their bags of artificial seawater. The researchers on board would help accelerate their growth during the mission. The astronauts worked around the clock in the Spacelab module, tucked inside Columbia's cargo bay. Over 9 days, they conducted dozens of experiments designed to reveal how microgravity affects living systems. The jellyfish were just one piece of a much larger scientific puzzle, but they would end up being one of the most talked about experiments from the entire mission. When Columbia finally touched down at Edwards Air Force Base in California on June 14, it had traveled 3.8 million miles and completed 146 orbits around our planet. The jellyfish on board had multiplied dramatically, and what researchers would discover about them would raise fascinating questions about life beyond Earth. So why jellyfish? It is a fair question. When Dorothy Spangenberg and her team set out to design this experiment, they had a specific goal in mind. They wanted to understand how living organisms develop in microgravity. But they faced a problem. Studying this in humans would be practically impossible. Human babies take years to develop, and the ethical concerns alone would stop any such project before it started. The team needed something different. They needed an organism that could go through its entire development cycle quickly, something they could observe from start to finish during a short space mission. Jellyfish turned out to be the perfect answer. These creatures develop from a polyp, which is essentially a stationary early life stage, into a free-swimming form called an aphyra in just 6 to 8 days. That timing mattered enormously. Space shuttle missions lasted roughly a week or 2, so researchers could actually watch the entire developmental process unfold in real time. They could observe how gravity-sensing structures formed in an environment where gravity was essentially absent. There was another advantage, too. The polyps could be induced to develop rapidly, which meant the team could produce thousands of test subjects from a relatively small starting group. This gave researchers a massive sample size to work with, something that would be impossible with larger or more complex animals. The sheer number of jellyfish meant the results would be statistically meaningful, rather than just anecdotal observations. What made this choice even more compelling was the biological similarity between jellyfish and humans. When it comes to sensing gravity, both use calcium-based crystals to detect orientation and movement. Humans have these structures in the inner ear. Jellyfish have similar structures called statoliths. If scientists could understand how these gravity sensors developed in space-born jellyfish, they might gain crucial insights into how humans born in microgravity would develop. It was a clever workaround to a question that could not be studied directly. The connection between humans and jellyfish might not seem obvious at first. After all, we have complex brains, hearts, and skeletons, while jellyfish are essentially floating bags of gelatin with no central nervous system to speak of. But buried deep within our anatomy is a structure that works almost exactly like what jellyfish have been using for hundreds of millions of years. Deep inside the human inner ear, we carry tiny crystals made of calcium carbonate. These are called otoliths, and they sit embedded in a gelatinous layer above specialized hair cells. When we tilt our heads or move, gravity pulls these crystals in different directions. The shifting weight bends the hair cells beneath them, and this mechanical motion gets translated into electrical signals that travel to our brains. Our nervous system interprets those signals to tell us which way is up, which way is down, and how we are oriented in space. Jellyfish accomplish almost exactly the same thing, but with different materials, and in a different location. At the base of their bell, arranged around the rim, are small sensory structures called rhopalia. Inside each one sits a crystal made of calcium sulfate, known as a statolith. When a jellyfish tilts or rotates in the water, gravity tugs at that crystal. It rolls or shifts slightly, brushing against hair cells that line the interior of the structure. Those hair cells then fire signals to the jellyfish nervous system, giving the animal a sense of its orientation. So here we have two completely different animals separated by vast evolutionary distance, both relying on heavy crystals and hair cells to solve the same fundamental problem. That shared solution is precisely what made jellyfish such an ideal stand-in for humans in these microgravity experiments. If space-born jellyfish could not develop a functioning gravity sensor, it raised serious questions about whether human children conceived and raised in orbit would face similar challenges. Once the shuttle reached orbit, the real work began. The jellyfish polyps were stored in bags filled with artificial seawater, carefully designed to keep them alive in the unusual environment of space. But the researchers needed more than just polyps. They needed jellyfish in their free-swimming medusa stage to properly study how gravity sensing develops. To make this happen, astronauts injected specific chemicals into the containers. These chemicals triggered the polyps to undergo their transformation, essentially speeding up the process that would normally unfold in the ocean. It was a delicate procedure carried out by trained hands in microgravity, where even simple tasks become complicated. What happened next surprised everyone. The jellyfish did not just develop, they multiplied over the course of roughly 9 days. Those original polyps proliferated at an astonishing rate. By the time Columbia was preparing to return home, there were approximately 60,000 jellyfish orbiting Earth. What started as a few thousand had become a swarm. The experiment carried a formal name that sounds like typical scientific jargon. It was called the effects of microgravity-induced weightlessness on Aurelia, Ephyra differentiation, and statolith synthesis. But behind that technical title was something deeply ambitious. The researchers were watching an entire generation of jellyfish develop their gravity-sensing structures in a place where gravity barely existed. Every pulse of those developing bells was a data point, and every statolith that formed was answering a question about how life adapts when the rules change. When the shuttle touched down in California after 9 days in orbit, researchers were eager to examine their subjects. The jellyfish had developed exactly as hoped during their time in microgravity. They had grown from polyps into fully formed medusae, and their calcium sulfate crystals had formed properly in many ways. The experiment appeared to be a complete success. But then the scientists tried to get them to swim. The differences between the space-born jellyfish and their Earth-born counterparts became immediately obvious. The control group jellyfish raised entirely on Earth swam with their typical rhythmic pulsing motion. They moved through the water with the easy grace you would expect. The space jellyfish, on the other hand, struggled dramatically. Scientists observed what they called pulsing abnormalities. The jellyfish seemed unable to coordinate their movements properly. They would pulse erratically, often tumbling, rather than swimming in any directed way. The researchers concluded that these jellyfish were experiencing something remarkably similar to severe vertigo, even though their gravity-sensing structures had developed correctly in terms of physical formation. Something fundamental had gone wrong. The crystals were there. The hair cells were there. The whole apparatus existed. But the jellyfish could not make sense of Earth's gravity. They had no idea which way was up. This finding carried profound implications. The problem was not in the structures themselves. The problem was in how the nervous system learns to interpret those signals. A jellyfish born in microgravity develops its entire sense of orientation without ever experiencing the pull of a planet. When you take that creature and drop it into Earth's gravity, its nervous system simply cannot cope. The signals coming from those crystals make no sense. The jellyfish is lost, disoriented, spinning through the water with no understanding of where it is or where it is going. The vertigo experienced by those space-born jellyfish raises an uncomfortable question for anyone dreaming of humanity's future among the stars. If a creature developed in microgravity cannot function properly when exposed to normal gravity, what would happen to a human child born in space? The implications are sobering. A child who spent their entire developmental period in weightlessness would likely face enormous challenges if they ever needed to visit Earth. Their vestibular system, the intricate balance mechanism in their inner ear, would have developed without ever learning which way is up. Walking, standing, even simply existing in Earth's gravity could become an overwhelming experience of disorientation and nausea. This creates a significant barrier for permanent human settlements beyond Earth. Imagine a colony on Mars, or a station orbiting a distant world. The adults who arrive there can adapt, because their balance systems formed under Earth's gravity. But their children would be different. They would be native to low-gravity environments, in ways their parents never could be. Those children might never be able to visit humanity's home planet without severe physical consequences. Research suggests that raising children off-world is not simply a matter of logistics or resources. It is a fundamental biological challenge that we do not yet know how to solve. Space tourism companies have begun talking about offering trips for families. But this research hints at risks we have barely begun to consider. A developing child might face consequences far more serious than the temporary motion sickness astronauts experience before humans attempt to establish permanent populations on the Moon, Mars, or anywhere else. We need to understand far more about how gravity shapes development. The jellyfish taught us that the problem is real now. We have to decide whether we can find a solution, or whether some boundaries should not be crossed until we truly understand what we are risking.